data science research lead for biodiversity and environmental sciences at the Berkeley Institute for Data Science, or BITSS. We've worked with them on lots of RT2, so we're excited to have BITSS back here again for RT2 2021. I don't, oh, there she is. Ciera's here. All right, so why don't we get right into it, and you can go ahead and share your screen.
We're gonna hear you.
How does that look?
Looks great.
Okay, so I'm going to discuss reproducibility for collaborative science. My name is Ciera Martinez. Hold on, let me move around this stuff really fast. I can't see the chat, so
we can, if you want us to, we can let you know if stuff is in there, or if you want to try to move stuff around on your screen and take a minute, I think that's also okay.
Yeah, let me just take 1 second and see if I can figure it out. Yeah, you guys could just let me know.
Sure.
I invite anyone to ask questions or interrupt me if something doesn't make sense. Yeah, I'll just continue on. So my name is Ciera Martinez. Like they said, I work at the Berkeley Institute of Data Science here at UC Berkeley, and I work primarily with biological and environmental data. For example, some of my research questions kind of are surrounded by how our environment and the organisms within it react to climate change. In addition to that, I train and establish data science reproducible workflows, and I'll go into a bit more about my background and where I'm coming from in my talk. So I'm going to give you a bird's eye view of designing a reproducible collaborative workflow that touches on kind of this whole RT2 roadmap without going into detail of any of it. So the keyword I'm going to stress here is designing. So while there are many different options, so there are a lot of different tools, a lot of different methodologies for building reproducible workflows. What I'm going to stress is that the overarching design principle is the people involved in the research projects. So it's you, your team, and your community. And this is going to be stressed throughout the entire talk. So my outline, the first part, I'm going to talk about the incentives and deterrents for reproducibility in general. That'll help you understand the really human aspect of trying to achieve reproducibility in your work. The part 2, I'm going to get to the nitty-gritty and advice on how to actually design these workflows. And then part 3 is really short, just reiterating some of the takeaways I hope you learn from the talk. So let's get started. So incentives and deterrents. So I read through your intro surveys, and a lot of you seem really well versed in the motivations for using reproducible tools, and a lot of you are pretty advanced on the tools and methodology. So throughout this talk, I'm going to leverage the knowledge in this room to understand. Oh, I forgot I'm going to use the I kind of need to see the chat so I can see what you guys are all saying. Does anyone know how I would see the chat?
So are you on, you're on full screen?
Yeah.
Would it, I'm wondering if.
Oh, wait, I think I found it. I found it.
Oh, great.
Okay. Okay, so my question for all of you is, what is the main motivation for learning reproducible workflows? And this is just for me to understand where you're coming from and for to see the wide variety of motivations where people are coming from. So just type it in the chat. Yeah, collaborating, learn from other people's work. Credibility, yeah. Improves the work. Responsibility. The ability to tweak, adjust, reproduce the workflow, yeah, definitely. Transparency. To remember what I did, yeah. That's like one of the main reasons I do that now. It's just to keep better notes. Yeah, there's a lot of transparency. Oh, avoid waste. That's interesting. Larissa, do you want to unmute and expand upon that?
Hi, can you hear me?
Yeah.
So hello, I'm Larissa. I'm talking from the south of Brazil, and talking about in the research group that I come from, we talk about how to be more transparent, how to share, how to be collaborative. And one of the things that comes up is to avoid waste in science, to avoid, for example, I'm doing some research A over here, and maybe my colleague is doing the same thing over there or has done it, and we don't speak, we don't know each other, but he found some similar results, or he seems that, oh, no, that's not going to work, or something like that. But we don't communicate, so I don't know what kind of troubles he's going through. Maybe I could adjust on my research, and so I wouldn't avoid wasting money, resources, time, and people. Motivation, that kind of, not just money, but all the thing that goes along among science and ways to make a better science. And I think the end of it is that to be more collaborative and transparent, so we can help each other to maybe build something better. I'm not sure if everybody got.
Yeah, that's a really good point. I always forget about that point, that if you put everything out there, everything that you did, you're less likely for your collaborator to redo exactly that, especially the early steps, which I'll talk about a lot during this talk. Those early exploratory steps where you're starting a research project and maybe you hit a dead end. If you're transparent about those dead ends, maybe your collaborator doesn't have to hit that dead end later. Yeah, so a lot of these are, in general, just making better science. So with that, I'm going to start defining my background and where I'm coming from in the open science and reproducibility space. So you learn a bit more about my motivations and how they've evolved over time. So I started out as a molecular biologist, fascinated with how leaves get their shape, a very esoteric biological question. And it involved a lot of alone time. So pipetting in the lab or staring at a microscope. And when I started my PhD, I had zero interest in programming, but I was forced to learn because turns out that pipetting and microscope work led to terabytes and terabytes of data. And I think that's a common theme in almost every domain right now, is that you go into it for the passion, and then all of a sudden you have a bunch of data and you're kind of forced to learn programming, reproducible methodology just to do good science. And so I learned programming for data analysis, like a lot of people learn, by asking around and Googling. There wasn't great courses like this when I started my PhD. And I ran into a lot of problems. There was no guides. There's a hardcore learning curve for tools. And yeah, like I said before, there's just no classes on reproducibility or even how to properly analyze data at the university level. And wherever I asked, people had a lot of strong opinions, and sometimes they were contradictory, and I didn't know what was right and what was wrong, or what I had to do versus what I needed to do. So when I started learning about computational reproducibility, my motivations were, one, I had a great fear that I would publish something that was incorrect. And this is kind of echoed with what you all were saying. I was extremely embarrassed of my sloppy workflow. So all the code, the data management, ability to reproduce results easily. And I was worried that I couldn't learn properly because of my sloppy workflow. And this came from just, you reach out to your colleagues who you think know what they're doing, and they look at your stuff and they're like, I can't believe you're not using Git. Like, I can't believe you're not commenting your code. And it comes from a very negative space. So I hear this from a lot of people. So the incentives for reproducibility often put this like big noble, it's what you should do. It's just how things are done. And it's often framed in a way that seems very intimidating. But that changed. So over time, my motivations changed and became more positive. And that was mostly for a lot of reasons, but they only became apparent when I started using the tools. This is especially with Git, which I know you all are learning today, but now my motivations are, it expanded my research along with my research questions. So learning these reproducible methods, all these tools and methodologies, it changed how I thought about research in general. My work was able to reach a wider audience, and I found community. So when you put your work out there, it reaches more people and it's easier to communicate what you did, and you find more people who are doing similar research. It allowed me to learn and perform research faster, which definitely was not the case when I started. It slowed down my productivity and my skill set. And this is a thing that I like to stress, especially for PhD students who may not want to be in the academic track. And I'll talk more about career motivations, but learning computational reproducible skills makes you more value and your career options open up. And that's especially important in academia these days. And the key here is that my motivations changed, and in this case for the better. And that might not always be the case, but it is the case that your motivations for doing reproducibility will change, and that's okay. The motivations for people on your team will change throughout time, and the motivations of your community will change throughout time. And that when you're designing reproducible workflows, they will change also in time based on these motivations. So reproducibility is really a fluid spectrum, and it's dictated by the people and the motivations for doing their work. So again, when you design with reproducibility in mind, you design for collaborative research. You create science that can be built off of. This better science component is kind of what unites everyone. The more I talk to people, like overall, everyone wants to do better science. And this really changed how I thought about my research. Like I was talking about before, my research questions got bigger. And also the world of researchers I could work with got larger. That reproducibility is really a language to work with people, work with more multidisciplinary scientists. So I see reproducibility and the tools I use to achieve as a language to work across different groups. And that's really what I do now at BITSS. I truly believe some of the biggest, most challenging questions right now in science require multidisciplinary frameworks and multidisciplinary researchers to ask. And one of the biggest questions and challenges that right now the world faces is how we're going to respond to climate change. So that's really what I've been focusing on. I went from, my very esoteric fascination with plant leaves to now I understand that, oh, wow, I can bring in the perspectives of all different types of scientists to make a difference in understanding of truly a challenging part of human history, which is how we're going to really react to this climate change. So as you can see, I am completely obsessed and convinced of the power of reproducible tools and methodologies, but that really doesn't tell the whole story. There are a lot of deterrents that face a lot of researchers, which is why reproducibility is not wholly adopted in every research community or with every researcher. So I mentioned a few at the beginning of the challenges. But what are some of the reasons, so this is another prompt, what are some of the reasons that make reproducibility a challenge? And you can say it's a challenge for yourself or that you've noticed in people you work with. And you can just type that in the chat window. Yeah, bringing along the team. I will hit on that. Definitely is a challenge. Yep. Learning curve is what I definitely hit when I started. Yeah, creating that pipeline, chaining together disparate tools. Even if you know the tools, how are you going to put it in a reproducible way? Ingmar says, I'm out. Oh, I lost it. I'm often the only coder in the work groups and collaborations. Many of my collaborators come from different traditions and go out and collect data. Yeah, we're going to talk a little bit more about that for sure. I went from being going to those people to being one of those people. So I've seen both sides. I'm not sure what is enough and not sure whether there are better ways to do that. That is a key one. Collaboration with the team. Yeah, lots of small decisions. Yeah, we're going to go definitely into detail with that. These are all, yeah, coworkers. Yeah, these are all reasons why I find reproducibility especially challenging. And it's repeated off of research in every domain I work with. One of the cool things about working at BITSS is it is a multidisciplinary institute. So we work with social scientists to neurobiologists to mathematicians. And this is repeated in every single field, these common deterrents. And another thing that is felt by a lot of communities is that they feel that their communities are the only ones dealing with it. Like another research community is well advanced in the subject. That's just certainly not the case. So why is computational reproducibility so challenging? And I've learned this mostly from trying to teach reproducibility over these 10 years. So I got really fascinated when I started to have these deterrents and trying to figure out my own workflow. And trying to teach it is also very difficult. And these are the three main reasons. So working openly and with reproducibility in mind takes time. And the key component to this is there's little incentive within academia for taking that time to do the work that makes it reproducible. Another really hard problem is every research project is unique. So it's unique in the skills and tools that you use. It's unique in the people you work with, their skills, what kind of tools they use. And it's unique in the community standards. So every community has a different standard. So it's hard to transfer that knowledge across domains. And lastly, data analysis workflows get confounded with a lot of software development workflows. So while you borrow a lot from computer science and software development, these are very different workflows. But the terminology that is used is often the same, and it's never really teased apart what the terms mean or what's synonymous, what's analogous, and what do you need to know versus what you want to know, or how you frame the whole landscape of what you're doing. You don't need to know computer science to make a reproducible workflow. but you couldn't tell that by how sometimes the terminology is used. So another thing is I want to mention is this bro open science, which is a really great hashtag if you want to look it up. It's that it's intimidating. There's a lot of people who are privileged and can take the time to do fully one-click reproducible science. And they kind of present it, dude, it's easy, just do it this way. And there's a real lack of empathy involved in how hard it truly is to do that, like one-click reproducibility. It's really hard, and the learning curve to technology can be steep. And the other thing is it evolves extremely fast. So even though someone figured out a perfect workflow for their perfect project, the technology will change in a year or two, and they'll have to relearn another workflow that becomes the new standard. So with that, you can see the wide spectrum of why people really want to and really don't want to delve deeply into reproducibility. Now I want to go into how would you design a workflow that works for your research, that works for your team, and that really fuels collaboration between the people involved doing that research. So I've been talking a lot about workflows, and I have not defined what that means. And a lot of times you see the word pipeline and workflow used interchangeably. So right now I want to just clarify how I'm using these terms. It's funny, if you look up like papers that describe workflows, they even put both words in the title. So pipeline, I'm describing it as a series of process and data analysis and data cleaning. This is usually linear in nature and can be programmatically defined by a computer. And it's described in relation to inputs and outputs. A series of processes involving, so the workflow is a series of processes involving humans navigating through performing these data analysis. So each step of the process can involve, so it's a mixture of code, machine automation, documentation, and then again, human intervention. And this is not necessarily linear in nature. So the key point here is that a workflow, and I think this is brought up for like earlier as a challenge, like putting all of those pieces together and communicating what you did is what I am talking about when I'm talking about workflow. So computational reproducibility, I hope at some point during this workshop, someone has talked about this very interesting definition or how people have been defining it over the years and have brought up the very confusing history of the usage of the word reproducibility, especially versus replicability. I put in this corner here a really cool paper that talks about reproducibility versus replicability, a brief history of confused terminology, which I found really interesting. To me, computational reproducibility, though, is just being really organized in execution. And being organized enough for someone to know exactly what you did when performing your work. So very fancy term with a very confusing history. I've just simplified it to you're just being really organized. So when you look at the leading tools and leading practices of reproducibility, you can see the main function of all of these tools is to be super organized enough to reproduce someone else's work. Version control is just being get whatever you're using for version control. It's just being organized on how something has changed through time. Data management, self-explanatory. Fair data, again, organizing how you present your data. Documentation, again, what did you do during this analysis? Making sure you're documenting all that, and then archiving, presenting it in a way that people can build off what you did. So what does a data analysis workflow really look like? So there are a few different ways to schematically understand that historically. Hadley Wickham of R fame famously illustrates this workflow where it's you import the data, you tidy it, then you kind of go through this section of your transforming, visualizing, modeling, and then you communicate it. Hillary Parker modified this by saying you're tidy. So this is the R world. Tidy means cleaning data. You clean the data, that takes most of the time. And then you kind of go through that cycle of trying to understand and analyze your data, and then you communicate your results. And the takeaway here is that both have in common as you over and over again, while you're exploring, you cycle kind of through this iteration. But me and two colleagues at the Berkeley Institute of Data Science, we really wanted to convey more. This isn't really what our workflows look like. So we got to work to build our own schematic of what reproducible data analysis looks like. And we created this schematic. And we wrote a paper on how this more accurately defines how workflows are really established and how to design an effective workflow for your team. And you can see in the corner there for a little part, I'm just going to kind of go into the schematic, but if you want to go into more depth, please read the paper that was just published in the spring in PLOS Biology. So we propose that data analysis workflow is defined and informed by a series of decisions. And the standards of reproducibility, and the standards being documentation, organization, tools, and code structure, this is a spectrum and becomes more stringent, the standards, as the project evolves through time. And that a research project often leads to dead ends and research projects throughout the entire lifetime of the project. We call it the ERP design workflow. Because you can also further separate this spectrum into the exploratory phase, in which your audience is yourself, the refinement phase, in which your immediate audience is your small team. That's when you start discussing kind of what is going on in your research. And then the polishing phase is where reproducibility becomes the most stringent because you're presenting it to your community. So let's start with the exploratory phase. So this is my favorite phase. I love to be in it. And it's also a phase where you hit a lot of dead ends. So you just kind of established your question and you're kind of exploring what tools actually work with the type of data. Is your data actually accurate? Is there something wrong with the data? Is the metadata you're borrowing from somewhere else? What's going on with that? Is the statistical analysis you're using, is it wrong? Is there outliers that make it impossible to ask your question? These are all dead ends that happen pretty rapidly when you start a project. You get hit with a dead end. And what's important here is that the reproducibility at this spectrum, you give yourself a little bit more freedom. You don't need to document all to full reproduce and to show your whole community exactly what you did. During the refinement phase, your audience is your small team. So depending on your field, in biology, for instance, your small team is usually your lab where you have lab meetings, but maybe your small team is just your advisor. Maybe your small team is just another person you talk to your research about. But because you're bringing that extra person into it, you have to start catering to how you communicate what you've been doing to that small team. And then the polishing phase is your community. And then again, this is different with every project. And this is where you want to show any products that reach beyond, that conversation level. And this is a really key phase because every phase has the potential to reach and produce a research product. And so research products, I don't know if you can see in the bottom here. So they're like a whole spectrum can come at any level during this workflow. And that it contributes intellectual education content to the research community. So it doesn't have to be a paper. It could be just documentation in general. It could be your project management, data management plan, sharing those. It could be a software tool. It could be a grant proposal. These are all things that are valuable for you, your team, and the community, and that can get you credit for working reproducibly. This helps get that credit part, that incentive that is kind of lost when people are trying to do this within academia. And it further motivates researchers to impose reproducible principles throughout the entire data analysis from the onset of the project. And so the schematic, there's a lot going on, which is I broke it up into pieces. There's this reproducibility line. So full reproducibility standards. So all the decisions, all the data cleaning, analysis, and computational environments you used, any tool you've used, how you've made those decisions. This only needs to be like in really good, fully like standard for your community, for the path, for the final product that communicates your scientific results. So a lot goes on in this whole research project and a lot of research products, but you only need to hit that like extreme level of reproducibility for the ones that communicates your results. And this gives you a little break on how intense you need to be on establishing those reproducible methodologies. So yeah, each of these little things refers to a product. And let's say this is a peer-reviewed paper. So you need to explain how you cleaned it, how you got to which statistical analysis you used, why you used them, what maybe even like supporting what tools you used to get to this final peer-reviewed paper. And this is I want to illustrate this is to give you that break that you need of trying to be absolutely every single part of your whole research project needing to be fully reproducible. So look at all these dead ends that happen in the exploratory phase. Let's say one of those dead ends involves cleaning a supplementary data set that in the end you just found unusable for whatever reason. Maybe you don't trust where it came from, or it just didn't say what the original paper told if you were doing a meta-analysis. Do you need to archive that weird dead end for public usage? Maybe you saw it's very valuable, and in the end, oh yeah, this is a really, let's say you're borrowing this, going along with that analogy, if you're borrowing someone else's data set and it doesn't say what it was supposed to say and it was published, maybe you want to publish a paper, and in that case, it wouldn't be a dead end. It would be you published something or you created a blog post that kind of points out what that does. But if you just go through it and it didn't say exactly what you're doing, or you don't trust it, you don't necessarily need to take all of those steps that it took to clean it and make it archivable and publish that code. And this again is to free you to be more nimble and creative during these early steps of a research project. And this is something that I kind of, again, am stressing throughout this. This exploratory phase is really important to get done as quickly as possible because you will hit so many dead ends. Let's see. So really fast, let's go into typing. You can answer by answering one of these questions. So does this schematic reflect how you perform your data analysis? how does it differ and how is it similar? Or you can talk about, and so label it 1 or 2. What are some dead ends of your past projects that didn't make it into the publication or didn't make it into a public facing product? And where do they live now? We'll give a second. Maybe a dead end is that your research question was not interesting, and therefore it's not valuable to the community. Okay, I'm going to, oh, yeah. Most of my dead ends probably in folders like dot, so hidden folders that are old. Oh yeah, I have a folder whenever I write anything that is just graveyard. And Fernanda says, yes, some of my work is more retrospective. So looking at a complete paper analysis and trying to make it reproducible. Oh, wow. So that is hard. Okay, I'm going to move on just because I see that we only have a few more minutes. I'm not going to be able to cover in detail all of this, but that I put these all in the slides for defining how to, the questions you can ask and define your team. So you can always come back to these slides to understand further. So design of your workflow is dictated by the audience and people and the research projects. And so I'm going to really quickly go into audience and people. So it's always guided by people. So you will need to define these people and discuss reproducibility with these people. So some questions you can ask are during the exploratory phase, and this is all in the paper, these little boxes on how to kind of guide this process, but who can read through our materials and understand our workflow? And then I give some advice on that. What materials do you think is worth continuing into the next phase? Who, for the refinement phase where it's your small team, who is this small team? What are your teammates' skill levels? Are there established standards within the team that need to be adopted or conflict with your exploratory phase? What tools can be agreed upon? What is the main takeaways of our findings? This will help you understand how to package an impactful research product. How will you be affected by the outcome of your work? Or who will be affected? Is your work accessible? What is, why is our work important for our domain specific field and for broader society? This helps you understand and prepare for the next polishing phase. And then for the community, who is the intended audience? Is it just one research field? Is it multiple fields? What is the next steps in the research? So who's going to build off of this research? Where do you publish? What are their standards? What's the journal standards for where you're going to publish? And why should you decide to build software tools? So I'm not going to go into this a lot in this talk, but you should see box 4. Oh, okay. I thought it ended at, oh, okay, good. Okay, I'm going to slow down a little bit then. So I just got a note that I have more time. I thought that we ended at the hour. So when do you expect research projects to be ready for a broader audience? And so I'm not going to go into tool development, but a lot of times a research product that is very valuable is creating a tool that gets you past those like intermediate steps, or it's a tool that you built just for your own research project, but you see the utility in using that tool beyond your research project. But yeah, read the paper. And when I use the word tool, I mean like a software piece or an R package or a Python package. Read if you should do that and what it means to your career. So for the polishing phase, a key point here is research projects can emerge at any time during the life cycle of the research project. So we talked about this line of reproducibility, but you really want to keep yourself and your team and what research projects to get credit for following this entire project workflow. So for instance, in the beginning, a lot of products in the exploratory phase, especially in my work, are turned into tutorials. So maybe it didn't work out. Let's say that this decision here is, should I use this software tool or this software tool? Or it could be, should I use R or should I use Python? And when I started to use R, I learned that, okay, these packages work, but A, B, and C packages didn't to a certain extent. But in the end, I learned how to use these packages. Create a tutorial, publish that tutorial, get credit for the work you did in this early exploratory phase. Put it out there, find community, and help the community. It's both being transparent and getting credit for this early work that, you know, may seem like a dead end, but in the end, you can get credit for it going further into that part. So you go through and you're kind of in the refinement phase. This is the time where you get to a part where you're seeing a clear path of results and a clear area of how you are going to publish what you've learned. You should start creating blog posts to kind of get feedback from the community on what you've discovered. And it also helps you be more transparent without adding it into the supplementary material. So it takes that supplementary material that is, or the metadata, and it brings it to the front. So you're both being transparent, but you're also helping the community understand what you've done. And of course, you're getting credit for being reproducible. That data management data management plans right now are publishable products. And they're publishable for a reason. They're hard to refine for particular products. Have other people learn how you managed your data management plan. And there are whole journals right now that you can publish those in, or use it in a grant proposal. Clean things up that not only make your project reproducible, but you get credit for that work. Have a workshop. You can see all of these things when it comes to the end. You learn a lot. And while in academia, the focus is on this traditional research paper, and it's true, if you're staying in academia, that traditional research paper is extremely valuable, but all of these other products are also extremely valuable for furthering your career, both in academia and beyond. So again, those research products are really your CV and your portfolio. So while you're aiming for full reproducibility, you can get credit for doing that by making sure that you publish or make public in any way to your community all of the different steps that you took to make that project reproducible. And this will also help motivate you and your team for working reproducibly. So why do you and your team want to use a specific method? So this was brought up before on a lot of the challenges for working reproducibly, is that maybe your team isn't on board, or maybe your team has drastically different skill levels than you. You're going to need to learn and compromise based on career aspirations. And what are those alternative research projects, and how do they fit with your career and your team's career aspirations? Maybe your PI doesn't care that you're doing reproducible work. All they care about is getting that paper out, but you have found that you want to go into industry, and that spending that extra time creating that computational tool is valuable. You need to be communicating that with your peers, and they need to be valuing your time and what you spend on it. And so I'll talk a little bit more about how to bring those conversations up at the very end. But I'm just going to quickly go through a few takeaways. So the main takeaway is that there is no single set of principles for performing reproducible research correctly. Don't believe it if someone tells you that. Each computational project carries its own context, from the scientific domain that you're in, to the software and methodology tools that you use, to the dynamics of a particular research team and how you define that team. You, your team, and your community motivations are really what design reproducibility workflows. It's not decided by some genius in your field who said this is the way to do it. You need to have conversations with your team. You need to really understand why you're doing the work you're doing. And you need to do a little bit of research on your community and what they ask of you. The research products, again, are decided by those motivations, and they can merge at any time during the life cycle of a research project. So again, get credit for the work you're doing by producing these research products. It's not just the final traditional paper. And establishing reproducibility standards while working on a project involves communication. So I'm going to end it right here with how we need to change our culture to really value reproducibility, but value the people and their time for doing this extra work that academia right now does not value as much as it should. So how would you do that? You reevaluate how your research meetings are structured, how you talk about your research. Don't just talk to your the people you work with about the cool result. Talk about the results that didn't work. Talk about your data management plan. Talk about what doesn't work using R versus Python. Talk about if you should actually be using Git. Do you need to use Git to write a paper? Is that maybe? But a lot of times the answer is no, there might be a better tool. Maybe Google Docs is just better. Include computational methodology in all of these discussions. So what statistics did you use? What didn't you use? Is it important to communicate that to the public? Probably. Do you communicate it in, how do you communicate it? In the paper, in the supplementary material, in the code that you publish? Include all of that in these discussions. Again, not just the cool results that you've gotten. You need to document the data management standards for your team. So as you're having these discussions, have a shared space for how to approach data management. Because what likely works for your team can be shared after you leave. There's a lot of turnover in academia. And if you document these things, they can be valuable for the next generation of colleagues in that particular space. Yeah, and talk about your career aspirations openly at every academic level. So talk to your interns, talk to your undergraduates about what they want from their career. ask yourself, ask your colleagues. And when you're deciding these data management plans, make sure that you're hitting the points, you're hitting these research products that get the people to where they need to go. Not everyone is going to be a professor. So doing reproducibility might be more than just being a perfect researcher. It might really help you get to a really good job that you love. Prioritize intermediate products that help your career goals. We've talked a lot about that. And encourage talking about reproducibility, documentation, and readable code on your team. Maybe you should start a code review or peer learning within your team, or start it within your community to get to normalize people seeing your workflows and your code. Everyone's workflows are messy. If we normalize the messiness and what does and does not work, that is really going to change culture. And what can really change culture is increasing your voice, or raising your voice. So it might seem awkward to kind of force people to talk about that. But again, reproducibility is decided by communities and by people and practiced by people. And you are a huge component of your community. So if your community is not up to where you want reproducibility standards to be, you need to start raising your voice from your small collaborators all the way up to community. And with that, I just want to acknowledge Sarah Stout and Valerie Vasquez, who are co-authors on this Principles for Data Intensive Workflows. And the BITSS Data Science Working Group, we have a lot of conversations surrounding all different aspects of data science and all these meta topics on how workflows are actually designed with people in mind. If you want to join that mailing list, there's a link right there. And then we're going to have a journal club here at Berkeley, which is going to be virtual. It's called Reproducibility, I think, because we're going to have tea. But I'm going to be their first journal club with the paper that I've been talking about throughout this presentation. So if you want to read the paper and have some more discussion, come to that. And I'm not sure if anyone has questions or how much time we have.